Hello, my name is Hannah Raczkowski, and I'm a master's student in the Department of Microbiology and Immunology at Western University in Ontario, Canada. And today I'll be talking a little bit about my research, looking at how the transcription factor Spi-C is dynamically regulated by external signals in B cells. One of the main focuses of my lab is looking at the transcriptional control of B cell development and B cell differentiation. And in particular, we look at the E26 transformation specific, or ETS transcription factor family. And more specifically, we look at the Spi subset of this family, which consists of 3 transcription factors, PU1, Spi-B, and Spi-C. Now, PU1 and Spi-B are very well characterized transcriptional activators, and they have a very important roles in B cell development as well as B cell function. These are also functionally redundant transcription factors, such that the absence of one can be partially or fully compensated by the presence of the other. Our lab has also shown previously that mice that are lacking these 2 transcription factors develop a B cell lineage leukemia. Now, my research focuses more on this other transcription factor, Spi-C, which is known to be a transcriptional repressor. And while it's much less well characterized than the other family members, it's been shown to have roles in antibody generating responses as well as certain stages of B cell development. Now, these 3 factors compete for binding to these GGAA motifs in the genome. And in doing so, they can regulate transcription of genes like Blink and Sick involved in B cell receptor signaling, BOC2, which is involved in germinal center formation and memory B cell differentiation, and other genes like NF-kB1. Now, most of the work in Spi-C has actually been done in the myeloid lineage, and these 3 major roles for Spi-C and the myeloid lineage have kind of emerged. The first on the left here being that Spi-C is required for the generation of red pulp macrophages. And these are a subset of macrophages that are present in the red pulp of the spleen, and they phagocytose senescent red blood cells and maintain iron homeostasis in the body. Now, it was shown that Spi-C becomes highly activated in monocytic precursors in response to the metabolite heme. And how this works is this transcription factor BOC1 is a constitutive repressor of Spi-C, but the binding of heme to BOC1 de-represses Spi-C and allows it to exert its role. In the middle here, it was shown that Spi-C is highly activated in response to TLR7 and TLR9 agonists. And this leads to the formation of these cells called inflammatory hemophagocytes. Now, these cells are very similar to red pulp macrophages in that they have a high phagocytic capacity, but they were shown to have a distinct phenotype. So these cells are present in inflammatory environments, and they were shown to have extremely high Spi-C expression. And on the right here, it was shown that Spi-C becomes highly activated in response to LPS, or sterile injury, in an NF-kB dependent pathway. And what Spi-C does here is it's actually able to convert macrophages in inflammatory environments to a more anti-inflammatory phenotype. And this is thought to modulate inflammatory responses and ensure that they don't get out of control. Now, in the B cell lineage, Spi-C has a couple main roles that have been discovered. On the left here, this is some work that's come out of our own lab where we showed that heme is able to activate Spi-C in B cells and that this promotes the generation of antibody secreting plasma cells. And we think that this is occurring in a BOC2 dependent pathway, very similar to the BOC1 dependent pathway that was shown to occur in the myeloid lineage. On the right here is a look at how Spi-C is involved in B cell development at this large pre-B to small pre-B cell stage. And how this works is RAG generates these double-stranded breaks that occur during small pre-B cell development in the Ig light chain genes. And this leads to the expression of Spi-C in an NF-kB2 dependent pathway. So what Spi-C does here is it binds to the promoter of genes like Sick and Blink, which are involved in B cell receptor signaling. And it actually does so by displacing its highly related family member PU1 and its complex. Now, what Spi-C is thought to do here when it binds to these promoters is that it blocks entry of these cells into the cell cycle. And this is thought to have 2 important roles. The first being that these cells with RAG-mediated double-stranded breaks obviously have a lot of severe genomic instability. So Spi-C acts to preserve the genomic integrity of these cells. As well, Spi-C's actions are thought to promote the differentiation of cells from this highly proliferative state that's characteristic of large pre-B cells into this more pro-differentiation state that occurs in the small pre-B cell stage. Our lab has also done some work to characterize the expression of Spi-C throughout B cell development and B cell differentiation. On the left, this is a look at some RT-qPCR data looking at Spi-C expression in 4 sorted populations. And we showed here that Spi-C is most highly expressed at the transitional B cell stage when these cells are exiting the bone marrow, but it's still expressed at detectable levels in pre-B or immature B cells as well as mature B cells. This is reanalysis of some RNA-seq data from another group where we looked at the expression of Spi-C in 4 mature or differentiated B cell populations. We found that Spi-C is highly expressed in follicular B cells, that it's downregulated upon entry into the germinal center, and that these cells highly express Spi-C as they reach these antibody-secreting fates, either in the form of plasma cells or plasmablasts. So this leads to the first objective, looking at the regulation of Spi-C by external signals in B cells. For this project, we hypothesized that Spi-C is dynamically regulated in response to external signals, and we believe that this is to regulate B cell function as well as B cell differentiation. Our aims for this part of the project were first to identify external signals that can regulate Spi-C expression in ex vivo culture of B cells, and next to investigate the relationship between Spi-C expression and B cell proliferation. We addressed these aims starting with extracting spleens from wild-type mice and enriching B cells using max CD43 depletion. From there, we went on to culture these cells with our various signals of interest and perform RT-qPCR for Spi-C expression, as well as cell counting. So this is a look at 3 different stimulatory conditions that we assessed, and these are all well-known proliferative signals in B cells. The first being LPS on the far left here, the middle anti-IgM antibodies, which stimulate B cells through their B cell receptor, as well as this CD40 ligand, which stimulates the co-receptor of B cells that interact with T follicular helper cells. And what we found was common between all 3 of these proliferative conditions is that Spi-C was robustly downregulated in response to all of these signals at most of the time points that were assessed. Now we looked at the expression at 24, 48, and 72 hours for each of these signals, and we found that all 3 of them significantly downregulated Spi-C. On the bottom here, this is a look at corresponding cell counts for these signals, and unsurprisingly, we found that these well-defined proliferative signals increase the B cell counts in culture over this time course that was assessed. So overall, from this, we concluded that proliferative signals robustly downregulate Spi-C in cultured primary B cells. We next went on to look at this cytokine mixture that contained BAFF, IL-4, and IL-5. And similar to in the last experiment, we performed RT-qPCR looking at Spi-C fold change and found that compared to freshly isolated B cells, BAFF, IL-4, and IL-5 caused a significant downregulation in Spi-C over time. Looking at our corresponding cell counts, we could see that B cell counts were significantly increased over the same time period, indicating that BAFF, IL-4, and IL-5 likely caused proliferation in culture. So at this point, we had seen this pattern where these signals that induce B cell proliferation were also downregulating Spi-C. So we began to ask ourselves what could interfere with B cell division. Now on the right here, this is a look at an experiment where we selected one time point and treated cells with either BAFF, IL-4, and IL-5, or the addition of this drug cytochalasin D. Now cytochalasin D is a potent inhibitor of actin polymerization and therefore causes a complete block of cell division. So what we found in this experiment, looking again at Spi-C fold change, was that BAFF, IL-4, and IL-5 once again downregulated Spi-C, but the addition of cytochalasin D was able to block this downregulation of Spi-C that we saw in these cells treated with BAFF, IL-4, and IL-5 alone, indicating that BAFF, IL-4, and IL-5 can reduce Spi-C expression, but that this is occurring in an actin polymerization dependent manner. Finally, we wanted to see what would happen when we just cultured B cells in complete media and assessed Spi-C expression. And we could see here that over the 72-hour time period, B cells significantly upregulated Spi-C, despite the fact that they were cultured in media alone. Looking at our corresponding cell counts, we could see that B cell counts were stable over the same time period. So overall, unstimulated B cells increased Spi-C expression over time. From this part of the project, we can conclude that Spi-C expression was significantly reduced in B cells following proliferative signaling through either of these 3 pathways here. that in B cells treated with BAFF-L4 and L5, preventing actin polymerization was able to partially block Spi-C downregulation, and that unstimulated B cells upregulate Spi-C over time. So at this point, we asked ourselves what factor or factors could be mediating this dynamic regulation of Spi-C in our B cells. So this led to the second part of my work. So we wanted to investigate the 3 regulatory regions of Spi-C that have been annotated, one of course being its promoter, as well as 2 regulatory regions that were previously defined. And in the top left here, we can see this anti-BOC2 ChIP-seq data that shows that BOC2 binds to these 2 previously described regulatory regions of Spi-C, about 40 kilobases upstream of its transcription start site. So we've deemed these regions of interest 1 and 2, and we'd like to assess whether BOC2 is able to modulate Spi-C transcription through binding to one or both of these sites. Just a little bit of a reminder that BOC2 is a lymphoid lineage transcriptional repressor, and it's known to promote processes including germinal center formation and memory B cell differentiation. And this is highly related to the transcription factor BOC1, which we know is a constitutive repressor of Spi-C in the myeloid lineage. And this is the pathway that we propose. The aims for this project: first, to assess activity of the Spi-C promoter in these 2 putative regulatory regions in B cells with luciferase assays. Next, to perform site-directed immunogenesis on binding sites of interest. And then validate activation of Spi-C transcription by NF-kB in B cells, which I'll touch on in just a moment. This is a schematic of the luciferase reporter vectors that were used in these experiments. So from top to bottom, the basic control vector. We clone the Spi-C promoter based on a track of its cross-species conservation, evolutionary conservation. And then each of these 2 regions of interest cloned upstream of that Spi-C promoter. And on the bottom here, these are our vectors with any sites of interest mutated, which we then went on to assess. So first, we found that transfection of the Spi-C promoter in the forward orientation into this Wee279 B cell line caused a significant increase in relative luciferase activity compared to the control or the promoter in the reverse orientation, indicating that the Spi-C promoter does have unidirectional activity in this B cell line. So from there, we were wondering what factor might be mediating some of this activity. And one of the things that we took note of is that we know that NF-kB, both the canonical and the non-canonical pathway, are able to highly activate Spi-C in the myeloid and the lymphoid lineage. So we thought that this was a possible candidate for the factor that might be initiating transcription through the Spi-C promoter. So we went on to use 2 types of software to look at any predicted NF-kB subunit binding sites within our cloned region of the promoter. First, we utilized this program called CIDR, and we could see that there were 2 predicted REL-B binding sites within our cloned region. And in particular, this one at 400 bases into our region was also identified using this ContraV3 program. And what was nice about the output of this is that not only were we able to see this predicted binding site at the same region in the promoter, but that we could see that there appeared to be this conservation of this subunit binding site across multiple species, indicating that this could quite possibly be an important binding site that mediates transcriptional activity. So what we wanted to do next was to mutate this binding site, and we mutated these 2 guanine residues to cytosines, which prevented binding of the NF-kB subunit to our promoter. And when we went on to transfect this mutated vector, we saw that there was a significant reduction in relative luciferase activity compared to the promoter in the forward orientation, indicating that this NF-kB binding site is required for full activity of the Spi-C promoter. At this point, we wanted to go look at our 2 regulatory regions that were identified by ChIP-seq. And on the left here, we started by transfecting either our basic control or a Spi-C promoter, as well as these 2 regions of interest cloned in either the forward or reverse orientation into this Wee279 B cell line that was previously used. And we found that when we transfected these regions of interest alone, there was no significant difference between these regions and the Spi-C promoter alone. However, we wanted to see if BOC2 was mediating any repression through interaction with these 2 sites. So we went on to acquire a MIGR1 BOC2 expression vector from Dr. Ji-Hee Yoon. And when we co-transfected with this Bach2 expression vector, we could see a significant reduction in relative luciferase activity when these regions of interest were co-transfected with the Bach2 vector, indicating that Bach2 expression reduces luciferase activity through interaction with these two regions of interest, and that Bach2 is likely repressing transcriptional activity in Spi-C. So from this part of the project, we can conclude that the Spi-C promoter has unidirectional activity in Wee279B cells. That mutation of a predicted NF-kB binding site reduced activity of the promoter in Wee279B cells, and that Bach2 represses transcription of Spi-C by interaction with two regulatory elements that were previously identified by ChIP-seq. This project is significant because first, Spi-C is emerging as a modulator of inflammatory responses, particularly in diseases marked by inflammation, such as ulcerative colitis. Spi-C induction could represent a novel therapeutic avenue for chronic inflammatory disease. And overall, the global identification of Spi-C target genes will enhance knowledge of the molecular mechanisms of immune responses, such as in the context of human vaccination and disease. I'd like to thank everyone who contributed to this project, as well as our funding sources, and thank you very much for listening.